Thank you so much for being here on Phacilitate TV. Can you quickly introduce yourself and your role?
Yeah, I'm Bella Neufeld. I'm the VP of Operations at Teknova.
Excellent. What does Teknova do?
We are a reagent manufacturer. So we specialize in both catalog products that are just off-the-shelf standard PBS, auger plates and such, all the way to highly custom reagents, so things that are very unique to whatever a customer might need.
Very good. Okay, so in the context of a lot of different new cell types coming out, some new viral vectors that are getting played with, even LNPs and whatnot, I'd be curious to learn what's kind of the process from first principles for making a customized reagent for these new applications?
Yeah, I think the first thing we have to discuss is sort of where we sit in the space today, which is a big shift, a paradigm shift to how we've done reagent manufacturing in the past. Because previously, we had these big blockbuster drugs, very large scale, 10,000 liter tanks required, and highly standardized platform technologies. And now we've completely shifted away to that, to, as you say, highly personalized medicine. And so not only does the industry need to keep up with that from a manufacturing perspective, but certainly the reagents that are required to do that have to shift as well. So really, what does that look like? It means much smaller scales, highly custom, but still high quality of standards. So being able to go all the way from process development, what we call research grade RUO, to commercialized therapies, which is more of that GMP. So spanning all of that.
Gotcha. And so yeah, if we're looking at these individualized therapies, like what are some of the first steps that you'll take for analyzing the modality and then developing the reagent that's going to best be suited for it?
Yeah, it turns out that a lot of these therapeutic companies are so focused on the science, as they should be, that maybe they haven't really considered all of the MSAT requirements for doing this tech transfer of the buffer manufacturing itself. And that's really where you need that partner to help think about those types of questions. Because to your point, every little step in the process is going to make an impact. And we know that because these are biological in nature, right, your starting materials are biological, often you have high variability, inherent variability in your manufacturing process. So the last thing you want is the buffer, the reagent, to somehow impact that variability. So trying to understand then, so what are the requirements from a buffer reagent perspective that can give you confidence that that's not going to play into that variability? So have you really thought about your QC specifications, your endotoxin levels, your pH levels, your conductivity? Have you really thought about what's the best format for the final situation that you're doing? Should it be in a bag? Does it need to be a fully filled bag? Or are you going to be doping in some additive later, so it should be only a half filled bag? What do you need on your certificate of analysis? What does your quality documentation look like to really cover all of your checks and balances for your quality system? So all of those nuances are things we would work through in sort of a tech transfer process to think about, just for the reagent. Now it sounds like a lot just for reagent, but it really is necessary.
Right. And so, what does that analytical panel look like when you're working with some sort of personalized new therapy? Is there a series of analytical tests that you're running to make sure? What does that tech stack look like?
Yeah, it's a tough question to answer because obviously we're not doing the end functional testing on your reagent. It's an unrealistic ask, and it wouldn't even really be representative of what you're doing. So then we have to come at it from a much more chemical approach and say, so what are the metrics that we can stand by to say, yes, indeed, this is exactly what we need? So there's more of the chemistry functional testing we might do, which is more like pH, conductivity, osmolality, density. Then there's the requirement for sterility, which is obviously really critical in this process. So getting USP 71 compendial sterility testing. And then there's more of the microbiology concerns, like are we concerned about DNases contamination or RNases contamination or endotoxin levels? And so all of that, we have sort of a standard panel that we could employ, but we could also talk about, again, for your process, like if you're really worried about endotoxin levels, then maybe we need to go even further down in the rabbit hole and say, is 0.5 EU per mil simply too high? We need to, drop it lower and lower and lower. And so that's all conversations we would have up front.
Gotcha. So shifting gears here a little bit, when we're now in this process of transforming manufacturing, a lot of different therapeutic developers started with more traditional manufacturing methods. A lot of new technologies even at this show are coming out that are really exciting with regards to maximize yields, to make product higher, but these are new approaches, these are new technologies. To what extent are you guys able to come in and help these therapy developers upgrade their process to fit in new reagents that are going to work in these new systems?
That's sort of where we sit today, right? Is you can either take, this is a challenge that drug developers will find themselves in. You can either take your existing technology that you know has worked for decades and try to morph it, slightly tweak it to then apply it to these very specific new modalities, or you can start from nothing. And we're seeing that across the board, not just from the platform for manufacturing, but also these technologies. So some of the technologies even here today, you can see they're actually a transformation of something that was used maybe for MABs, and we're tweaking it to now use it for a different modality.
Right.
Versus creating something from scratch. Both of those are challenging. Both have their pros and cons. We're here for it. Whichever route goes, you go down that path. But it's definitely one of the challenges I think we're seeing in the field now is you can't just change an existing modality or platform, and you really can't start from scratch. And of course, nobody's really sharing their secret sauce because it doesn't make sense to do that. So everybody's sort of trying to figure it out at the same time, which means it's a very difficult space to live in.
Makes sense. So yeah, then if I'm a therapeutic developer, I'm looking for a manufacturing partner, for a reagent developer. What are some of the key qualities that I should be looking for that makes for a good partner?
So because of this paradigm shift that we've talked about, obviously scale is a huge impact. You have to find someone that can go to smaller scales than what we've seen previously. And I'm talking like small, like 8 1-liter bottles at a GMP level. Because sometimes that's what we need to do to start with, knowing we're going to grow substantially. But in the beginning, we just have to have that confidence at that level run, right? Because these batches are much smaller than what we've thought about for previous. And then custom is the other piece. So again, previously we have a standardized platform process, which includes reagents. And so you can just pump those out at 10,000 liter scales over and over again. Now what we've seen is these slight nuances, these slight differences in the reagent actually have profound impacts on the purity or even the reproducibility. So having a partner that can do that level of customization is really critical. And then of course, obviously you need to be fast, right? Everybody wants speed.
Yeah.
I'd say the last piece there though is the quality. So how do you have a QMS, a quality management system that can span all of this scale from 8-liter bottles to 200-liter bags, and that wide variability of customization? How do you have a quality management system that can span all of that? And those are sort of the 3 pieces I think you really need to think about.
Gotcha. So let's dive into that a little bit, right? I'm in the process of developing a custom reagent. I want to get this done pretty quickly, but obviously you want to ensure quality as well. So what would you, I guess a better way to explain, to talk about this would be, how can you best kind of match the need for speed while also making sure that quality of custom reagents stays really high?
Probably a number of different approaches. I can tell you the approach that Teknova has taken is what we'd call modular manufacturing. So what this looks like is you can basically take at any point in the manufacturing process, all the way from raw material choice, like which raw material you're going to choose and then what QC test you're going to do to that, to formulating, to mixing, to titrating, to dispensing, sterilizing, to your formulation, final formulation, your container, all of those pieces, we call it plug and play. So you can take any of that out and put something in that's unique to whatever customer requirement there may be, as opposed to saying we have our standard platform end to end and you have to somehow fit into that. That's where it becomes really tricky to move quickly. So you can do it, but now we have to set up an entirely new line, a new process to fit that. So instead we've said, hey, you want this raw material, we can swap it in there. You want this QC test, we can swap it in there. And you can just do that throughout the entire process so that you can get on the floor really, really quickly.
Makes sense. I'd be curious, are there any other, what would you say are some of the big challenges what are some of the big challenges that some of these different therapeutic developers are currently experiencing when it comes to making these reagents?
Yeah, I mean, I think it's part of the question is really back to how does anyone really know how to get high purity at a reproducible process? And that is inherently what we're dealing with today. And again, if nobody's really sharing that information, that's, we kind of find ourselves in this position where we're all trying to figure out at once. I think this lack of reproducibility is huge. Again, I take it back to we're dealing with biological starting components. So of course it's going to be really challenging as you move through that process. So from our perspective, we want to get the buffer right. We want to make sure that's one less thing you have to worry about in your highly variable DOE that you're running. So if you're trying to see these nuances, what is the impact of purity? But those nuances could be impacted by having a conductivity that's a little bit higher than it was last run.
Right.
Then you really have no insight into whether that's a real data point that you can rely on.
Very good. So I'm a therapeutic developer. I'm looking to develop custom reagents. Why should I go with Teknova?
Well, we have our modular manufacturing platform that really relies on these three pillars that we talked about. So we need scale. We need to be go all the way from RUO to GMP,
Yep.
but also from 8-liter bottles to 200-liter bags. We need it to be customizable. So everything from the formulation to the C of A to the QC testing to the finished good container. And we need really high quality rigor, really high quality standards. So one of the ways that we've been able to accomplish that at a GMP level is to take a bracketed approach to all of our validation, to say we're going to bracket 1-liter bags all the way up to 200-liter bags, and that will cover everything in between. So by doing this bracket for every situation, again, we can move really quickly. We can plug and play things in. So if you come with a brand new formulation that we've never seen in a new container, all of these pieces, we can quickly get it on the floor.
Fantastic. And I guess I'd be curious to get your thoughts about how do you see reagent production and customization evolving as we go into the second half of the 2020s?
Yeah, it's an interesting question because it obviously could go one of two ways. Either things do become more standardized and platformed, and everybody starts to understand like these are the critical quality attributes that matter for a buffer, and therefore we're gonna capitalize on that and move in that sort of MABS direction. Or I think actually what we're seeing is things are becoming even more personalized and specialized, and therefore we won't ever be able to really standardize it. And so you have to just be ready to move on the fly and say, hey, today conductivity didn't matter, tomorrow it turns out it really does. Or that little add of poloxamer make all the difference in the world for our aggregation input.
Right.
So I tend to think it's not going to be standardized anytime soon, again, as we move more and more to personalized medicine, but you never know.
Well, thank you so much for coming on.
Thank you.